So this was the last slide of the previous presentation. And just to recap, what we've got here is an industrial fermenter. And within the industrial fermenter, what we are doing is we are producing the human insulin product. And we're doing that with genetically modified bacteria. And the genetically modified bacteria have recombinant DNA. Recombinant DNA is a combination of the bacterial DNA and then the human insulin gene. The transformed bacteria are capable of doing transcription and translation and construct the human insulin product. The advantages of doing this is that if you are diabetic, human insulin is much, much more specific than the predecessor, which was to use pig insulin. And pig insulin was essentially extracted from carcasses of pigs and smashing up pig pancreases, and the purification process was very, very difficult. And on top of that, pig insulin is not exactly the same as human insulin, so the efficacy is different. Another associated problem with using pig insulin is for certain religious groups like Jews and Muslims who don't use pig products. So this technology and the advancement of science is very helpful for diabetics. So what I want to underscore here is this idea that the bacteria is transformed, and it's transformed by using a plasmid to insert the DNA from the human is inserted into a plasmid. So the plasmid is referred to as being the vector in this case. And a vector is any DNA molecule which can be used as a vehicle to artificially insert foreign genetic material into a host cell. And when that foreign material is inserted into the host cell will express the particular gene and produce a particular protein. Another vector, other than the use of a plasmid, which is on your course, is to use a virus. So let's just pause and think about what we know about viruses. Viruses are not, strictly speaking, thought to be living. They're not biologically alive because they cannot do the characteristics of living things, Mrs. H. Gren, without their DNA being inserted into a host cell. So viruses are naturally occurring organisms which are, well, not really organisms, but they're going to insert their DNA into a host cell, and that host cell is then going to express the viral DNA, make viral proteins which can be reassembled to make new viruses, which then go on to infect other host cells. Now, if you think about this idea of a virus specifically attacking a particular host, the host is usually fairly specific, then viruses can also attack specifically bacteria. And so there's a virus which is referred to as a bacteriophage, and a bacteriophage is a vector which inserts its viral DNA into bacterial cells. And what we can see here in the yellow is the DNA, and it's going to insert it into the bacterial cell here, which this is then going to express those that viral DNA. Now, if we can manipulate what we have within the viral DNA, then again, we can have a genetically modified bacteria. So viruses can be used as vectors in addition to this idea of what we've already met, taking out the plasmid and then cutting it, inserting the DNA and placing it back in. Now, genetically modified bacteria are used industrially to produce all sorts of products, and there are 6 examples which are screened up here. And what I'd like you to do is to pause the video in a moment and take a look at this section of the textbook and make some notes on each one of these. And as you go through, just signpost your way back to what we already know about particular things. So go back, when we're looking at human insulin, we've talked about how this is produced in quite some detail, but revisit what insulin actually does within the body. Remember, it's there to balance the glucose levels within your blood. It works alongside glucagon. Make sure that you're clear with what that insulin molecule is actually doing in the body. Enzymes we talked about here and down here, and enzymes, remember everything that we know about enzymes, active sites, lowering the activation energy, etc. When you look at 3 and 4, They are both involving hormones, and remember what you know about hormones. Hormones are released from endocrine glands. They are transferred around the body within the bloodstream, and they target specific sites to have an impact. So insulin is an example of a hormone. Then revisit what you know about vaccination programs, and remember that when you do a vaccination, you are going to deliberately insert an attenuated form of a bacteria or a virus into the bloodstream. And what you can do, rather than use the whole organism, is that you can inject an antigen into that person. The person then is going to raise an immune response. They're going to produce specific antibodies against a particular antigen. So as you're making notes on all of these 6 examples of genetically modified bacteria, make sure that you're connecting it with previous related work that you've done. So here we need to revisit what we already know about transgenic. Remember that transgenic is a term that describes an organism that contains and expresses DNA from an unrelated organism. So we've got transgenic bacteria in the previous slides as examples. And here I want to draw your attention to the idea that we're looking at genetically modifying plants. And when we genetically modify plants, what we will then go on to do is make lots and lots of those genetically modified plants. So you've got to connect this with what we know about micropropagation techniques. Now, here we've got an organism called Agrobacterium tumefaciens. And Agrobacterium tumefaciens is a naturally occurring bacteria which infects plants specifically. And like all bacteria, it's going to have plasmids. It's quite easy to take out the plasmid, to cut it with restriction enzyme, and then to insert a known DNA that you want. So we can insert any kind of DNA that has got some kind of desirable effect that we're interested in incorporating ultimately into the plant. But first of all, we put it into the plasmid, we put that recombinant DNA back into the agrobacterium, and we allow the agrobacterium to then infect the plant. The plant will then have within it this particular plasmid, which will be expressed to produce a protein with some kind of desirable trait. And to just illustrate this point, let's consider this example of BT corn. And BT corn is this one here. It's nice and healthy, as you can see, compared to this one, which is the wild type. Now, BT corn has got inserted into it DNA, which comes from a bacterium which is called Bacillus thuringiensis, hence the name BT. And this particular gene will produce a protein which is toxic to particular insect pests, and in particular the European cork borer, which is responsible for the damage that you can see here. So the European cork borer is going to infect this plant, and it's going to cause damage. It's going to reduce yield. Remember everything that we talked about at the beginning of this unit. Whereas if the European cork borer tries to attack this genetically modified version of the corn, the BT corn, then it will start to eat the toxin, and it doesn't get very far, as you can see. Now, is this a good thing? Well, you could say that it's a good thing because it means that if you've got this BT corn here, you reduce the need to use a pesticide. And remember that pesticides can be damaging to the environment. They can target species that are not the pest itself, like pollinating insects. Also, the pesticides can hang about in the environment for longer periods of time. Pesticides can also be problematic because they can move through the food chain if they're ingested. So we've met the idea that pesticides are bad. So does that mean that genetically modified food is good? Well, not necessarily. One thing to consider is if you've got all plant species, flowering plant species rather, which corn is an example, can produce pollen. And the pollen produced from this particular plant is going to contain that BT gene. Now, if the pollen then starts to breed with a related corn species, but not this particular kind of corn, what you're going to get is the BT gene being infected into plants that were not intended to be infected with the BT gene, not a crop plant. That can cause problems if it finds its way into a weed. Then what you've got is as a weed that's producing BT corn, the BT gene, and that weed can flourish when you don't want it to be flourishing. So some exam questions, as you already know, are going to ask you to evaluate a particular instance. It could be that paper 2, question number 1, talks about some kind of genetically modified plant and asks you to evaluate whether this genetically modified plant is a good or a bad thing. On this next slide here, there's one error which I want to pull your attention into, but this is useful for you to read and to consider. So why would we genetically modify plants? Well, we will genetically modify them possibly for some kind of nutritional value. We can have a higher yield of protein, higher yield of vitamins. We can actually genetically modify also to have the plant produce a particular vaccine that we want. We can make crop plants which have got reduced allergens, which is important, obviously, if it's going to be consumed. We also can produce crops which have got characteristics which allow them to be hardy. For example, they could be drought resistant, frost resistant, salt resistant, all the things that we spoke about previously in terms of artificial selection. But make sure that you appreciate that here we're not doing artificial selection. We are deliberately introducing a foreign gene to get this particular trait. Now here there's an error. It says genetically modified crops that can produce herbicides to kill pests. Now just remember that herbicides are targeting specifically weeds. So when you look at this pest here, that's not very clear, I don't think, and I didn't change this. I should have done, but the herbicide is targeting a weed. So what we've got here are genetically modified crops that if you spray herbicides all over that crop is not going to cause damage to your actual crop, but it's going to kill all the weeds which are growing in and around. And again, what can happen here is that the genetically modified crop can produce pollen. That pollen can then cross-pollinate with other wild-type plants, and wild-type plants now become herbicide resistant. And if that wild-type plant is a weed, it's going to be herbicide resistant, which means that it's potentially going to become a real problem. So what I'd like you to do again is to pause the video here, to think and consider all of these benefits. Also think of the risks that are associated and the drawbacks that are associated with genetic modification, and just prepare yourself for an evaluation kind of question within a past paper.